Morning, biologist. I hope all is well in the world of social distancing and remote learning. Today, now that our lab reports have been submitted, we're going to take some time to refocus ourselves on the other content of this unit by working on our study tool, which perhaps is something we have forgotten about in all the craziness, but good news, you will not be forgetting about it today. So I have the study tool terms up over here on my screen, and I just want to take some time to walk through some of the things we've talked about so far and make sure we're all on the same page. So in regards to things we've covered so far, these first three sections, we've only covered part of this tweaking gene expression section, so I would say you might want to save that section till after spring break when we actually cover that. But as of yesterday, we have also completed this last section, making mutations. Okay, because it's been a little bit of time since we've actually talked about some of this beginning stuff because the focus has been our microarray lab, I want to remind us of our transcription translation POGL that we completed before we left for remote learning. So many of the terms that are included on the study tool are going to be found in this POGL. Many of the pictures in this POGL are also going to be extremely helpful when reminding yourself about the processes of transcription and translation. So I just want to take a few moments, walk through each of the processes, pointing out a few of the key terms that you'll need to be explaining on your study tool. So central dogma of biology states that DNA's message gets converted to RNA's message, which gets converted to proteins. And this happens through the two processes of transcription and translation. The first process, being transcription, is pictured here. And in terms of components that carry out transcription, it's pretty simple. It's this one enzyme that we see right here, RNA polymerase. And this enzyme can really do the job of all of the enzymes that we saw in DNA replication. So it starts by splitting apart the double helix of DNA. And then it bonds to the promoter region at the start of a gene. This signals to the RNA polymerase, here's the beginning of a gene, here's where you start transcribing. As it moves along the DNA template strand, which is always in the direction of 3 to 5, so the RNA polymerase is moving from 3 to 5, adding complementary RNA nucleotides. So hopefully we remember that that RNA nucleotide will have a ribose sugar instead of a deoxyribose sugar. It will have a uracil instead of a thymine. So it starts building these complementary nucleotides, verifying that it's the correct nucleotide by hydrogen bonding. That hydrogen bonding being weak is very temporary, and the string of RNA falls off the DNA almost as quickly as it's synthesized. So as RNA polymerase is moving 3 to 5, it's building 5 to 3, a new RNA strand, until it reaches the terminating sequence, which tells it, hey, this is the end of a gene, you could fall off, and we could all carry on with our lives. Then we get this lovely messenger RNA strand, mRNA. Okay. This mRNA strand is not quite ready to leave the nucleus. It's going to have to go through a little bit of processing in a eukaryote. So this processing only occurs in a eukaryote because in a prokaryote, this RNA is already in the cytoplasm. It's already ready to go. So the mRNA in a eukaryote is going to get a cap to help it find the ribosome. It's going to get a tail to protect it from enzyme degradation, and it is going to get the introns spliced out or removed. So when you splice out introns, you basically cut the phosphodiester bonds and remove those nucleotides, ligating together the exons that surround it. Exons are part of the mRNA that actually gets expressed. It's the part that actually codes for the protons. The introns are not needed to create the proper protein sequence. So once our mRNA is all mature, with its cap, its tail, and without its introns, it's going to leave the nucleus through a pore and find a ribosome. Now, the ribosome is out in the cytoplasm floating around, so the cap will help it find the ribosome, and the ribosome will bond to the mRNA right around the location of the cap so that the first thing present in the ribosome is the start codon. And a codon is a word that refers to 3 nucleotides on the mRNA strand that code for an amino acid. And we're able to figure out which amino acid a codon codes for by using the genetic code table, which is pictured up here. So that first start codon, AUG, besides telling the ribosome it's time to start, also codes for the amino acid methionine. And that amino acid methionine is going to find its way to the ribosome thanks to a tRNA molecule. And the tRNA molecule knows it's in the right place at the right time if its anticodon is complementary to the codon on the mRNA. So here we see our codon AUG is able to bond to the anticodon UAC. But an important thing to note is that this tRNA is carrying methionine. That's because AUG codes for methionine. You never want to look up the anticodon on the genetic code table. You always want to look up the codon. So the tRNA complementary to the first codon comes in, followed by the tRNA complementary to the second codon. Once both tRNAs are present in the ribosome is going to help facilitate the dehydration synthesis reaction and form a peptide bond between the 2 amino acids. At that point in time, the ribosome is going to shift down the mRNA molecule, forcing the first tRNA molecule to leave, and the second tRNA molecule is going to be left holding the 2 amino acids until the third comes in, at which point it'll pass off its amino acids to the third. From here, the ribosome will move again, this tRNA will exit, and this guy will be holding the whole amino acid chain. This sort of pass the amino acid chain is going to happen until the ribosome reaches a stop codon. At that point in time, the stop codon will be recognized by a release factor. So it's not a tRNA molecule, and it's a protein that just bonds to the ribosome and signals, hey, let go of that amino acid chain. And here we go, is our amino acid chain. We do not yet have a functional protein. This amino acid chain needs to fold in order for that to happen. Very often this folding will occur in the rough endoplasmic reticulum. The folded protein will then be passed to the Golgi body so it can be shipped to its proper location for functioning. These steps I just walked through can be referred to as initiation, elongation, and termination. And the genetic code we used earlier is something you will always have available to you on tests and quizzes. And there's a few important things to note about this genetic code. A, there's 64 different codons, which seems like a lot being that there's only 20 different amino acids. But this is an incredible thing because we have multiple codons for a single amino acid. For instance, look at leucine. Leucine has 6 codons that code for it. So what that means is if this A accidentally gets mutated into a G, my protein is unaffected. And this is because the genetic code is redundant thanks to a phenomenon known as the wobble base, meaning when this tRNA bonds to this mRNA, it's less concerned with what's in this third position. If there's not a perfect match here, the tRNA might still drop off its amino acid. And in the long run, that's a beneficial thing to ensure that the proteins we build have the proper primary sequence so that they can have the proper secondary, tertiary, and quaternary sequence, allowing for the proper function. All right, guys, so that's basically a roundup of these first 3 sections here. So I'm hoping those things clear some stuff up for you or re-clarify some things for you and allow you to create a very useful study tool. You still have the option of doing a digital version or a paper version. If you do a paper version, you're going to have to take some pictures of it and drop it into your Google Drive folder. Your completed study tool is not going to be due until after spring break, but at the end of the day today, I'm going to be checking that you made some progress in these first 3 sections as well as this last section. I do not want to see blank documents. I do not want to see like 1 or 2 words defined. In theory, you have an hour to work on this. So you should have some serious progress underway by the end of today. All right, I'll be available on Zoom at 1 o'clock if you have any more questions or you want to clarify anything further. And I hope to talk to you soon. Have a good day, guys.